Hi. Thanks for staying with us. Núria will join later. And meanwhile, we can start with Lukasz Lamza.
Hello.
Lukasz, hi. Thank you for being here. So you are a special case here. Actually, you have several special cases here because the conference kind of mixed disciplines, mixed fields. Well, you're one of them. So you not only study biological questions, you're also a journalist, you told me, you also teach. So can you tell a bit more about these two parts of your life, more humanitarian, more science, and how do they influence your view of biology?
Sure. Essentially, I'm interested in storytelling. This is how I see my job as a journalist, and this is also how I see my job as a scientist. I'm always interested in finding a story. This is what journalists do, right? Something is happening, and I try to find the story that's going to get people interested. So what's happening and why is it interesting? This is very crucial to me, to find a reason why something is interesting. So in case in this of what we're doing here, for instance, when I look at history of life, I try to find certain patterns and certain events that I think would interest people. So this is essentially, I try to please people in both of my jobs. So I don't know if it's normal, if it's a typical way of doing biology. I don't think, right? It's not.
No, I don't think so.
But when you think about it, I mean, you have scientific conferences, right? And you would have 50 people presenting. And people will go back doing the work, and some things that they have heard will stick with them. Like the kefir thing that I think so far has probably won the conference, right? Because why? Because it's a good story, right? Yeah. This is and I mean, there's fantastic research here, but still it's just too much for people to proceed. So I still think that we should think in those terms, like, what am I actually bringing here that will stick with people? And my answer is, well, a good story.
Actually, it's a really good approach, because now you said that you try to please people. I think mostly in research we try to please ourselves because we're curious about what we study, and I think it's more selfish field. But indeed, I think recently, well, always we talk about that, but recently this topic is getting more and more popular. There should be more science communication, because there is like a wall between scientists and non-scientists, no? And if you break this wall, it will be better for everyone. For non-scientists, because, well, they know what we're doing, and we're doing it mostly on their taxes, no? And for us also to recruit more people so they understand us better, and the communication knowledge will spread more. So I think it's a really good approach.
Thank you. Can I say something?
Yes.
Or I actually teach science communication to scientists, and on next Tuesday, just when I go back from Barcelona to Poland, I'm going to have a course with academics, teaching them science communication. And this is usually the first thing that I say. Okay, so show me your research, all right? So tell me, what's the interesting story in this paper? It's like, you're supposed to interest a random person in 30 seconds.
So
tell me your story, right? So why is it hot? Why is it interesting? And many people are surprised. Oh, well, no, you know, what I do is not really that interesting. You know, they get defensive.
This one kindness.
But I said, no, you're doing this. I know that you're interested in it because you're a scientist. We all love our, you know, I have never met a scientist who would actually, under questioning, say that, no, it's not interesting. Everybody thinks that deep inside it's interesting, but scientists are afraid that when they go out to people, they won't find it interesting. So I guess the trick is to, as I said, try to please people. It's like you meet people in a bar, or you just meet with your friends or family, and they're asking what you do, and you're scrambling in your mind to try to present what you're doing in an interesting way. I think we should be doing the same scientific conference and in the papers as well.
No, it's definitely a good idea to teach science communication to scientists. I agree. Okay, Núria, thanks for joining. So I asked Lukasz that he is a journalist and a researcher. So this is like two fields and how they influence them. A bit similar question to you. So before you work in the Iñaki group, and your organism was Capsaspora, Philisteria. Now you changed to Thibault group and you work with conoflagellates. So thanks. So of course it's the same question, the same approach, but it's completely different groups of organisms. So do you feel that it's changed also your research life and your approach to see things?
No, great question. Well, first of all, welcome everyone. Great, happy to be here. Fantastic meeting. Yeah, I think that the fact, I mean, a very good friend of mine once told me, usually when you transition in research, during your research careers, many times you can change your question. Some other times you can change the model. Other times you can change like the approach, no? And then sometimes it's very challenging because you change like the 3 things or 2 things at once. So in my case, I think that having like the same background question really helped me to be more open-minded about how we approach transitions to multicellularity in this case. But I think that a key difference for me was the fact that that the models that we use, which in this case are quanoflagellates, we can directly go to the field and find them again. And that was like a big difference for me on how I understand research and how I understand or how I interpret, let's say, what I find in the lab, no? Because now I can go back to the field and have more of an ecological perspective about what might be happening, no, in the wild.
Yeah, with Quanos is definitely more chance to get a grip of ecology. With Capsa, it's no idea, no?
Yeah, exactly.
Okay, so then I will ask you my second question now, because it will be a continuation of the story. So, yeah, we have this organism, we have the model, the approach, and there are several other approaches. We learn about some today also, and yesterday, and the day before yesterday. So, but it feels like we need to get further, no? We don't have all the tools. Like science never have all the tools, all the ideas. So what do you think would be the next? What we are missing? We need something like, what would be next step, which will be future for the multicellularity research?
Wow.
I know.
It's a fantastic question as well. I think that a key challenge for us is to be able to ask the right questions at the right time. Now we have many tools and we need to start to apply them. So for example, there was a huge breadth of single cell technologies emerging. I think that it would be fantastic if we can start applying those technologies into our organisms to get more of a hint on how they might be cooperating, integrating different information and maybe differentiating spatially, temporarily, and how that works. I think that putting ecology in the spotlight, I think that that's a key thing, and I think that now with experimental evolution we're starting to do that, no? Like incorporating different selective pressures to try to see outcomes, ask then what's next, no? What's like the, for example, fitness advantage or what might happen next, and I think that then like the long-term question, which is the functional side, which is I think something that it is still lacking, and it's been lacking at least in our community for over the past 20 years. It has been a dream of our community founders and the people and the generations that are following. So having the genetic tools and the right experimental conditions to be able to functionally test the function of key genes and the interaction between the internal environment of the cell at the cellular and maybe colonial level, as well as the external environment, no, like chemical cues, signaling, communication. I think that would be next challenges that we will all be facing.
Yeah, that's definitely we need to do that because it became too easy to sequence.
Exactly.
So we did too many omics.
Exactly.
Yeah.
But as Maya said, I think that also the challenge and the mission at the same time is to have fun and in between.
Yeah, okay. Selfish part again. The research. Yeah. Thank you, Núria. Yeah. And so also regarding to that, I really liked to see in your talk that there is much more to multicellularity than we usually think of. There is things which we don't know. It's like they are like multicellular. They have function differentiation, compartmentalization, but they are one cell. So I really like that this idea that we need to look at them also, and there are many of them. It's like not exception, no? So, and related to that, you said that you were searching for topics which would be interesting. How do you search them? What is your approach, and how did you find this topic?
I think in this case I was fascinated by the sheer richness of protist lifestyles and life cycles, and especially the ones mentioned in 2 or 3 papers that were written 30 or 100 years ago, and nobody is willing to study them, but they were described in quite a lot of detail. But I think what we're kind of lacking there's hope too, is actual study of the diversity of these creatures, and I have huge hopes for the environmental, metabarcoding and now culturing these creatures. I'm waiting always for these untapped potentials in environmental sequences. So my method actually of finding interesting stories at the, you carry a tree of life would be to find creatures that it looks like from the evolutionary patterns that something happened over there, but we have like 1 or 2 species and we see that there's something going on, like central helids. They are not studied very much, but there's dinoflagellates. There's a couple of weird things at the basis of the dinozoan tree of life, and there's a whole lot of environmental sequences that just have been named, marine sequence number 72. So what I do is I try to very carefully see what's going on in terms of finding these creatures actually and culturing them and describing them morphologically, because I think this is a huge untapped potential. So this is kind of my method of doing what I do. I read others people's work mostly, but looking specifically for those places that you see by the 1 or 2 species that have been described that there's something interesting going on, like a simple form of multicellularity, but we only have 1 species and a shrubs and trees of uncultured lineages, and when we culture them, this is going to be big.
It's like journalistic research to the science approach.
Maybe.
Yeah. Cool. Thank you so much. It was fun. Yeah. Thanks for joining and see you later. Bye.
Thank you.